cover three major aspects. One of it would be the major innovations happening in the therapeutics intervention space. And the second one would be, we would briefly touch base on AI and its role in new biotherapeutics, but very briefly and later we'll also talk about how drugs been discovered in one part of the world, how they can be used to treat diseases in every other part of the world so that they don't have to go through the discovery process all over again. And lastly, we'll close with how lifestyle interventions can prevent diseases, or even combinational therapies can help improve the quality of life of patients. Without further ado, I would like to first introduce Anjali from Sudo Biosciences. She'll explain to us about how the drug discovery process works, right from the design, biochemistry of the molecule, till it's all through the clinical trials and the drug release into the market, because she has wide experience on that for many years.
Hi everyone, thanks for coming here. And I would like to go over, I'm a trained chemist by training. I think people probably don't know what chemist does in pharma industry. The chemists at the start, medicinal chemists make drugs, molecules. And they interrogate not just the chemistry molecules, they interrogate the biology, the pharmacology, pharmacokinetics, and toxicology to build in a molecule. And so I'll walk through how the drug discovery and development works, which is virtually be identical either for small molecule or a biologics, a large molecule. You decide about a target and a disease that you want to go after. What is that biological target that you want to attack and you think that is going to help that disease population? And from that, once you identify the target, you go through target validation to make sure this target really is going to be disease modified. And once you have done that, you kind of have to think about what is that molecule that can, or an antibody, what can that modulate and affect efficacy of that molecule? And that's where a lot of chemistry comes, and either you utilize a high throughput screening, or with the computational technologies now, you can do virtual screening of billion molecules with your target of interest. And when you are screening, you're looking for how these molecules interact with that in a 3D dimensional, that protein of interest. And that's where a lot of work goes on when you start designing molecules. It's like, I call medicinal chemistry as an art. It's like, how do you design molecules? What are the key interactions with your protein of interest? And if it's the crystal structure or the 3D structure is not available, thanks to DeepMind, now having AlphaFold, where you can build the protein. So a lot of this AI technology now is coming into drug discovery, helping. So when you go through, design your molecule, then that is called, you identify a hit. Then hit goes through identification of the lead molecule. Then you do the lead optimization. In the phase of lead optimization, you have to, as a chemist, you have to do a lot of multi-parameter optimization. You want to make sure the molecule has the right potency for the target. It is selective for the target. It doesn't hit anything else because that can lead to adverse event and toxicities. You want to get the right pharmacokinetic properties, that the compound can get good bioavailability, good absorption, all those parameters, and then having good pharmacodynamic properties, and that's what is called lead optimization. And once the lead optimization is done, then you do all the preclinical work, in vitro, in vivo, going from rodent to non-rodent, testing all the properties of the molecule, and then doing in vivo efficacy studies. When I say in vivo efficacy studies, that's the animal models that you build for the disease that you're going after. And that's where you look at the efficacy in here, and you also then do some toxicity studies, because you want to have a therapeutic window. FDA is not going to let you take a drug if you don't show very good therapeutic window, that this is the lowest, and at 10x of the plasma concentration or 20x of the plasma concentration, this compound is still safe. So once we do all that, we get the efficacy, toxicity done. You have to do 4 weeks of tox in rodent and non-rodent, and that is when it becomes an IND package. PK, PD, CMC. Now you have to scale up, make a process of your compound, scale up your compound, and then you file an IND, scale up, GMP, tox, and then next phase is phase 1. You go into healthy volunteers, single ascending dose, multiple ascending dose. Then you go into phase 2, your proof of concept studies, where you're testing your patient population somewhere between 200 to 500 patients. That's where you see the efficacy of your molecule and also learn about either any adverse events. And then you go into phase 3, which is a big patient population, ranging from 1,000 to 5,000 patients trial. And after all that, you submit an IND if it's a small molecule, or a BLA if it's a biologics. And this whole process it takes 10 to 15 years process. So I would stop here. Definitely in the regulatory, there are different drug approval processes. And then we can talk about that later, because I want to give others time to talk.
Yeah.
But that's a nutshell of start to finish of a drug discovery process.
Yeah. So as Anjali explained, targeting one protein of interest takes about 15 years. So now we'll hear how we can leverage the innovations in medical technology to kind of develop antibodies that can target 2 or 3 targets from Sachdev.
Hello? Yeah, thanks. It was refreshing to hear some real intelligence, not artificial intelligence. No, it is a complicated process, and I don't think drug development will ever come easy. It's just the way it is. My group works on antibodies, but what we're excited about is why do one target when you can do multiple targets? I think the real future is hitting 2 things, 3 things, 4 things. And there you do need all the knowledge. Nobody can read the literature anymore. It's just too much literature. You need it organized in a way. And we're working with Pradeep and Sam over here on Think Bio. And what we're looking at is, let's just remine literature to find connections that we missed and make an antibody that hits 2 things or 3 things. And I think that's, ironically, the future is in the past. Many targets that failed because one target just wasn't good enough. If you can hit 2 things, and we've been making agonists, which brings things together, so you actually stimulate cells rather than turning them off. So I don't call it AI, I call it literature mining. There's a lot in the literature that we can dig back into and find new ways of combining stuff that's already, maybe 20, 30 years old, but wasn't targeted the right way in the past.
And when we're targeting 2 or 3 proteins, how do we make sure there are not side effects throughout the body when we're designing these drugs? Sachdev. When we're, the side effects aspect of it.
Well, interestingly, there's a fallacy that hitting more things necessarily gives more side effects. You can actually, we've had examples where we add an arm to an antibody, which relieves toxicity because you direct it where it needs to go. So, and that's the big game changer. Making it more complex can actually make it safer because you're exactly sending it where it needs to go. We made a drug that's in the clinic now with Merck, an eye therapy, and it has to bind 3 things at the same time, but then it corrects a defect. So there's this misunderstanding that making a drug that hits more things will become more toxic. Yes, if you randomly throw a cocktail together, of course that's going to be toxic, but you can actually make drugs that are bigger, hit more things, and paradoxically become safer because they're doing they're going to the right place. They're not going randomly. So that's exciting to me. I think toxicity can be reduced by more complex drugs.
Now I would like to hear from Shalabh about how existing drugs from one particular disease can be used to treat other diseases, with specific examples on what you have worked on in that space.
Hi, good afternoon, everyone. My name is Shalabh Gupta. By way of background, I'm a trained physician. I practiced medicine for a decade. I have a finance background, worked on Wall Street as a stock analyst, worked at Genentech in strategy group, and founder CEO of this company that we started a few years ago. I will answer that question, but most importantly, my best day in my last several years of professional career was this past Monday. We announced a drug that got accepted by the FDA for a PDUFA date, which is an approval date, which agency has given us for June of next year. We've gone through a lot of these processes that Anjali pointed out, but I want to talk about a slightly different way to think about what we do. I don't have Anjali's background. I do not have your background, but I have a different background. And I think about the drug development as a complicated puzzle, which we are trying to solve, and we all have different roles to play. I'm fortunate to have met people like Anjali. What you realize that developing a drug, think about this as a 5 different types of risk. Anjali talked about chemistry risk. We can talk about antibody drug conjugate as a biology risk. So if you think about 5 different risk factors, so there's a chemistry risk, biology risk, clinical risk, regulatory risk, commercial risk. These are all risks in any drug development that you do. What matters is that how you think about risk. Developing a drug is a scientific pursuit, but at the end of the day, if people are not going to give you the money to develop the drug, it wouldn't really matter. And eventually, if the drug doesn't have a place in the market, it wouldn't matter. So just going back to this part, I started this company with no asset, no money. So the question is, how do you start a biotech company with no asset? Or typically, people think about biotechnology, and we were having this conversation earlier, as a marathon. I don't think we can run marathon. Most of us, if you are an able-bodied person, you can technically run a marathon, but you will have to be trained for it. Most of us, if do proper training, we can at least run a mile or 2, and that is what I call relay race. So I wanted to break down marathon into relay race. I'm not an inventor. I have a garage, but I always tell people that a biotechnology company, if somebody develops something in their garage, you probably don't want to fund it. It's not worth developing it. But what I found that there are technologies and drugs that are being developed in other companies. Some of them, not because they're bad drugs or bad technologies, but they sit in those companies because they are not necessarily strategically aligned. More specifically, but more directly, in our example, there was a drug that another company had developed over 10 years, and it spent over $25 million. The company that was developing the drug, they became very focused in cancer, oncology, and they were publicly traded companies, so therefore their investors didn't want them to do anything other than oncology. And we were building a company focused on nephrology or kidney diseases, so I was able to acquire the drug from them with no cash, with a small equity upfront. After acquiring the drug, which was completed a phase 1 clinical trial, a lot of things that you will hear commonality. We think about what actually is the development pathway, and for us, the shortest, the most direct pathway that is most efficient in cash and also time is the key to be able to develop it. I'll fast forward. I don't want to talk throughout this is a panel discussion, but in an abbreviated form, after acquiring the drug, we were able to shorten the pathway of regulatory approval, make it from phase 1, that is what was completed, run 2 small trials and file for approval, which we did a few months ago, and we just recently, as I mentioned to you earlier, this past Monday, we announced that FDA has accepted our drug for approval and review process. It's not approval, it's a review, but this is the last step that happens, which gets the drug to approval. They give you a timeline, and FDA says, based on what you provided, we think you have sufficient information that we can review and approve your drug. We can talk more about it.
I would request the panelists to hold the mic closer, please, so that we can hear it properly. Yeah. Now we'll hear from Sam Santoshi, like having spent 20 years in the software industry and 15 years in this space, how the role of genomics in drug discovery has been.
Thank you. I'm Sam Santosh. As she mentioned, I was in the software industry initially, but about 15 years back, I moved into the genomics when I figured out that biology has become digital, and we have access to the, the source code of life, getting the genomic sequence. So that was in 2008, 2009 time when I set up the first genome sequencing lab in India, and then it's a good journey over the last 18 years. In building up different companies in India focusing on genomics. But coming to the human genomics in particular, I mean, we all know, I mean, our code, our genes are the basic for, reasons for all the way we look, our all attributes, our intelligence, and, as well as our susceptibility to disease. So India, fortunately or unfortunately, is a place where it's a good place to find new drug targets. Why? Because India has one of the most ancient population. Because once people moved out of Africa, India was one of the first places to get populated about 40 to 50,000 years back. And then there have been multiple migrations from various parts of the world to India. But about a couple of thousand years back, India somehow, for good or for bad, developed the caste system by which people separated into groups and would not intermarry. So India currently has about 4,500 very distinct population groups which has interbred among themselves for the last couple of thousand years. This has obviously led to a lot of increase in diseases. So that's why I said the good and the bad. There's a major challenge. India has about 5 to 6% of children born with a serious inherited disease. So that is sort of like the population. India makes about 26 million children a year. The sort of the population of Australia, and a year. And out of that, about when you say 5 to 6% children, that's about more than 1 million children born with a serious inherited disease. So those are the challenges. But on the other side, we have got a lot of unique variants, unique genetic variants, which can help us find new targets, and also understand also why the reasons of disease susceptibility. We all still haven't figured out why some people get some disease, while others don't, even in the same house, even when they share most of the genome. So your genes, your microbiome, and then maybe some luck factor, but at the same time, there are very significant genetic reasons by which you get a disease. And as Dr. Sidhu was saying here, when you go for multiple targeted treatments also, you need to get the more genetic information, because all targeted therapies depend on the basic genetic information that you have. So all this genetic information, as we collect more and more from patients, will help us definitely in advancing new therapies.
Yeah. And one more question for you. So the new therapies have been slower to come to India compared to the rest of the world. How can we expedite the process of new therapies coming to India?
Yeah, India is a country with a lot of challenges, right? I mean, we have a very large population. Like I said, we have huge diseases, huge disease burden, and we don't have enough resources for many things. So India could not develop or be up in the front of research. In the last maybe 20, 30 years, India has not stepped up where it could have. India has a good, strong pharmaceutical industry, for example, because we are very good in making generic drugs. So India is even called the pharma capital of the world, and we are very good in, for example, in getting vaccines out, for AIDS and for many other things. We could get very cheap vaccines, doing it the job much better than most of the big pharma in the world. But unfortunately, when it comes to new therapies, in the last maybe 20, 30 years, India completely missed the boat. There has not been a single antibody that has come out from India. So that is on one side. Second side is that India is poor. So of the 100-odd antibodies that is currently approved in the world, less than 25 antibodies are available in India. So the challenge is big. Behind in technology, very poor to afford new medicines, lack of many insurance and lack of other facilities to diagnose. So the challenge are multifold. But like new technologies from people like Dr. Sidhu, those can be good things if India is able to bring them quickly to the Indian market.
Thank you. So now we'll hear the most exciting thing from Dr. Yatin about personalized medicine.
Good afternoon, all. So I think you heard from all the other panelists about drug development and how expensive it is and the time duration that is taken. Tens of millions of dollars, if not hundreds, and takes 15 odd years to develop a new compound. The other side of the coin is if you start looking solely at clinical studies, so which of these compounds actually make into clinical trials and get to approval, it's a very small percentage that actually makes it and gets approved by the FDA, right? Depending on the disease area, it's 3 to 5%, 7%. So what we do as a company is we focus on understanding why it is that such few drugs that have passed the tolerability and have passed the safety margins, so they are showing good performance in phase 1. They perhaps even have shown good efficacy in phase 2, but you get to phase 3 and you flunk the study, and it's goes into the waste bin. So apart from all the wasted dollars, there's also the challenge that these drugs that actually didn't make to the clinic were perhaps really good drugs for certain patient population, which is now going to suffer from lack of that specific drug in the market. So what we've done is we've come up with, much like Sam was saying, we've come up with a software model of a human cell, which can then be personalized to reflect each specific patient's disease. So that before the patient is actually injected or administered the drug, you can make a prediction of how that drug is going to perform in that patient, purely from an efficacy perspective. We don't make any projections or predictions of toxicity or safety and tolerability, purely from an efficacy perspective. So, so you can then be running these trials not on the patient in the clinic, but you can run those in software, and only once you have sufficient confidence can you then go into the clinic with that particular drug. So that's the general idea. And then the way we monetize this is we take this to the clinic to guide clinicians on which therapies might be applicable or might be ideal for their patients. And clearly in doing so, we have to go well beyond the FDA companion diagnostics, because companion diagnostics already provide you some level of guidance. So you've got to go beyond that. And then the second application and use case, which is on the pharma side, is we buy out compounds that have stalled in phase 3, that have either failed phase 3 or for whatever other reason have stalled in phase 3. And we rerun the phase 3 clinical trials and get them back into the clinic. As of now, we are focused purely on oncology and immunology. Those are the only two arenas. But over time, this could be applied to other complex diseases as well. So that's a nutshell of what we do.
Can you also please touch base on the combinational therapies, combination of drugs?
Right. So as far as the modeling is concerned and as far as the therapies are concerned, we are agnostic to whether it's a monotherapy, combination therapy. In fact, it could be any number of drugs that can be applied to the model and to the patient. So in terms of rescuing a compound that has failed, we look at the scenario at 3 different levels of risk. The lowest level of risk is rerun the study, exact same study that Big Pharma had run. Most of the compounds we buy are from Big Pharma. So rerun the exact same study that Big Pharma had run. However, with one change, which is you don't admit all the patients, you only admit those patients that we believe are likely to respond. So same combination, same investigational agent, simply patient selection. The second level of analysis we can also do is, in addition to patient selection, we often change the combination partner or the companion molecule, because often we find that pharma has picked the wrong companion molecule. So we change that, which of course raises the bar a little bit, because there will be no phase 2 run for that study, so we got to go back, rerun phase 2 as a proof of concept, and then come back to phase 3. And the most difficult one is one where pharma may have just picked the wrong tissue type, the wrong disease area. So instead of renal cancer, lung may have been a better target. in which case, if they've not done sufficient phase 1, you've got to go all the way back to phase 1. So that's how we take a look at it, but it's independent of monotherapy or combination.
Thank you. So before we move to the second segment, we'll take just 2 questions from the audience, and then we'll come back to our panelists. Does anyone have any questions for this panelist?
So I have a question for the industry practitioners or drug developers, both on the antibody side and small molecule. We talked about all these digital tools coming online. Can you give some specific examples of how they may have helped in an actual drug that's been developed? And particularly, you know, with DeepMind and all these structures that are available for proteins, can you just completely avoid the crystallography and structural information and go directly to a drug? Or what are best practices today? What's the state of the art? Both small molecule and the antibody side.
So, no, I think you can't get away from crystallography. I think it's still your target, the ligand in the protein crystallography is still
Anjali.
the key. Sorry. Okay. Yeah, so if I understood your question, you were saying, can you bypass crystallography? I would say no. I still, in all the work we have been doing, we still use structure-based drug design quite a bit, which means you get the crystal structure of the protein, and then you dock with your ligand with it to really understand what are the key interactions of the molecule, especially when you're designing, either you want to be best in class or first in class. If you're doing a small fragment-based discovery to get best in class, first in class, then you start a small molecule, some small fragment, dock it into your crystal structure, and then start building. So I don't think it's going away. AlphaFold is also only for when you don't have crystal structure. You're going to build AlphaFold models to help you initially, kind of thing, but eventually you would need your own crystal structure. But a lot of time, lot of medicinal chemists don't sometimes even use the structure. It's a lot of intuition. Okay, you looked at the structure and say, based on crystal structure of this molecule, if I modify at this position or that position, we can build our own new chemical entity and have the affect the target.
So is it fair to say that it's mostly a research academic tool, not really an industry tool so far?
Yeah.
Yeah, thank you. Now we'll move on to the AI segment. Sorry.
I was going to comment on, I respectfully disagree, but because we're in a different field, I know for small molecules, structure is critical, but yes, the strength of antibodies has been, you don't need the structure. I like AlphaFold is fun to look at, but I have over 100 PDB entries of my own. But for us, always been journalism. You develop Herceptin, I didn't develop it, but it took 10 years to get the structure of Herceptin. So antibodies have been a wonderful means of targeting things that you could never fully understand until you targeted them. They're big, they're complicated, but they're easy to make because they're genetically engineered.
So when you say you do a literature search, what do you really look for there for your antibody design?
Pardon?
When you say you do a literature search for digital sort of mining, what do you really look for?
Well, we have a saying in my lab, 10 billion antibodies can never be wrong. We have a library of 10 billion. You have to be very good at the methods, which are screening, selecting. But the wonderful thing is we've made antibodies to things which we may never have structures of, but they're perfectly fine drugs. That is the advantage of antibody engineering. It's why there's 100 approved antibodies in only 20 years, right? Because you can make them, dare I say, without understanding them.
So this is two different things. You're trying to do biologics, antibody versus small molecules. You still get a lot more small molecules than biologics from FDA. So antibody, still the small molecule is the area where you get those approvals from FDA.
So Anjali, if we can hear from you about how we are leveraging AI to optimize the multi-parameters during the drug discovery process. Can you please use the microphone?
Yeah, definitely. I have, you think old-timers, but I think AI is here to be embraced. And where I think, at least in the drug discovery phase where I've started understanding and embracing is utilizing it early discovery when I'm doing a lot more multi-parameter optimization. There are a lot of good AI tools that are built on so much data. As a human, I can't get that. So I think it's helping from that angle, looking at multi-parameter optimization and getting the right pharmacokinetics, absorption, permeability, that kind of parameters, kind of helping that. And also a lot of virtual libraries and those screening tools that are there from the computational are trying to bring it in, which will decrease the amount of time as a medicinal chemist, a chemist to make or design, because a computational can look at a bigger space than I as a human being can. So I think AI is there to help that. And I always consider AI and all these tools as one of the tools in my toolbox. It's like, how am I going to use to make me more efficient? And what can I do, use machine to do that job instead of me spending, which I couldn't spend, even if I have 24 hours, I need a lot more hours to spend. So I think that's where you'll see the timelines on discovering a molecule will shorten. Look, that's where I see an impact. And then the second impact I see in patient selection is a big one. I think that was talked about. So where I see why drugs are failing. We see phase 2 efficacies and stuff like that, and still fail in phase 3. It's like, what was wrong? Was the target wrong? The compound was wrong? Or the patient selection? So I think that's where I see the impact, especially in the drug discovery timeline. We will decrease that timeline in getting the right molecule. I'm talking about small molecule right now. I'm trying to focus, but that's where we'll make a difference.
Thank you. If Sang can talk about how AI is helping us advance genomics, especially to handle the large data to pinpoint to specific new targets.
Yeah, sure. I mean, genomics is data, and so it's one of the most appropriate fields to use AI on. And so, in fact, before even all this, the new surge of interest in AI happened, AI, as in this old avatars in machine learning and other ways, has been extensively used in genomics. The whole field of bioinformatics relied extensively on AI tools, and they are been embracing all the new technologies as they come up. So more importantly, in finding for new targets or for new discoveries is not just the genomic data. It will be more important to get all the associated clinical data, environmental data, lifestyle data, if you're talking about humans. Similar microbiome data, all that you will need along with the genomic data to make any sense of that and to correlate whatever findings that you're seeing to some other outcomes or some measures that you can take to alleviate that. So discovery will come from those type of efforts.
This is a question for any of the panelists to take. How do you think AI has been a bane for the drug discovery? Any limitations that we need to address before leveraging AI?
Oh, you're asking the right person. I worry about AI because many of the greatest discoveries have dumb people doing stupid stuff. And you build programs that keep telling you, don't do this, don't do that. Here's 50 rules. Nobody imagined 150 kilodalton protein like Herceptin could be produced at scale. And so I worry that AI is going to simply replicate rules that stop people from doing weird stuff. Science is not about rules. There's a great Bob Dylan line. He said, Bob, how do you break all the rules? He said, I don't see any rules. I think we could end up in a position where people stop doing stuff because the program said, nah, don't try that, it didn't work. 82 times. It could work the 83rd time.
Thank you. So we'll switch gears a little bit, and if we can talk about, for example, most of the new therapies are being invented here, as Sam has mentioned, but how do we make sure the drugs invented in one part of the world are being leveraged across the globe so that they don't have to go through all the process again? Any of you can take this.
This is a difficult question for me. We do provide services on the clinical side in other regions of the world, not just in the US. But one of the key challenges we find is some of the more basic and fundamental technologies needed from a clinical perspective. Genomics, for example, Sam talked about that. Simply not prevalent enough in the rest of the world. And I'm not just talking about India, but several of the other developed world as well. One might argue that these are not prevalent in the US in the clinic, but if they're not prevalent in the US, they're barely available in the rest of the world. So certainly from a clinical perspective, there's a big lag from discovery to the products actually being available in the clinic. On the drug development side and the drug distribution side, what we do find is, especially in non-oncology, Gilead is a great example who's making their compounds available in India. In fact, the day these compounds are available, hepatitis is a good example. The day these compounds are available in the US, they were also made available in India, generally through a licensing method and mechanism. So there are some examples of compounds being made available and drugs being made available, but not yet widespread enough. I don't know if others have to add.
I think we decided very early on that we are a small company, we can develop drugs everywhere in the world, but our company is focused on kidney diseases, which are universal. Not to take 15 minutes to talk about kidney disease, but obesity, diabetes, they're all precursor to chronic kidney disease, and it's increasing as we see the society demographics, not just in the US, all over the world. So we decided to partner with the regional companies very early on, and at least in our therapeutic area, the local FDAs, so we have partner program in China with Chinese FDA, we have partner program South Korea with Korean FDA. They do believe that you don't have to replicate the trial. You do have to provide the dossier, and they all, whether they directly say or indirectly, they all tend to follow FDA's premise, but it is possible to not have to go through the clinical trial in every geography regionally. The only caveat is that sometimes, for example, our drug was developed in the US. It has only US patient population. Chinese FDA says that we have no data on Chinese ethnic patients, so you may have to run a small clinical trial in the regional geography. So that is possible, but you can leverage US data. Depends on therapeutic category, but you can leverage it globally.
Thank you. So a closing question for any of the panelists. So at what stage do we engage the FDA and other regulatory agencies so that we make sure we are putting our efforts in the right direction, right from picking the molecule to all the way to taking the drug into the market?
I'll try to answer this question. So I think, many times there are all these people like Anjali, they help to innovate molecules, but I'm not a chemist. Definitely antibody drug conjugate is not my thing. I think from a very different vantage point. There are two reasons drug either succeed or fail, but the number one reason is universal. Doesn't matter what modality you're developing. The first rule of medicine is do no harm. So the first rule, critical rule, is understanding the side effect profile. Before a drug gets approved, the number one cause is many drugs fail because they cause toxicity. Part of the things we are talking about, phase 1, phase 2, phase 3, a 1 in 1,000 patient having a heart attack seems very small, but the same when you start to give it to 10 million people, now you have thousands of patients dying from heart attack. What I just gave you an example is the reason why a drug like Vioxx, which is a COX-2 inhibitor, was pulled out of the market. So the difference between human lives and software is that 1 in 1,000 app, when you download, it crashes your iPhone, you reset it, but that's not the same in human being. Drug development, in our case, in our drug, 99.98% of the drug never gets absorbed in the body. It gets excreted in the stool. The drug we have in our portfolio, the drug we developed, it is a nanoparticle-based technology, but we were able to communicate with FDA that our drug is the same drug that FDA had approved several years ago, so that's why expedited pathway. So the point I would like to remind that, look, on one hand, there are many ways to get to the finish line, but at the end of the day, agency or we as a drug developer or industry care about making safe drugs, because if the safety is a question mark, then we can't succeed, and then comes the efficacy. And I think you heard many different ways to get to the finish line. So if you have a communication with FDA, as much as we believe they're out there to tell you what not to do, they actually do want to have more drugs approved. And I'm in Anjali's camp that, I've been always reminded that nobody really cares of a small molecule. I have been only in a small molecule. I think if the drug works, it helps patient, you will figure out a business case. It's not that everything has to be antibody-drug conjugate. I don't mean that they don't have a role to play, but there is a role for all modalities. What matters is we provide solution to the patients.
So we are purely clinically focused and completely modality agnostic. So we work with small molecules, biologics, CAR-Ts, all cell therapies, gene therapies, all kinds of things. And to your question about how soon to talk to the FDA, I think I'll echo what Shalabh just said. We found the FDA to be very constructive in all our discussions. So we talk to them as early as we can. Of course, we are mostly focused on phase 3s and then occasionally phase 2s. So we find talking to them, the more you talk to them, the better off you are. There is a bad reputation in the press that the FDA generally has, but our experience has been quite different. They tend to be very cooperative and tend to be quite helpful in providing guidance, including suggesting rather innovative study designs. Occasionally, we'll be having an internal debate of should we even propose a specific design because it might be a little bit aggressive. And we'll go in to talk to the FDA and we'll find that they'll propose it to us, even when we were a bit hesitant. So I'm going to say their fearsome reputation is not quite justified. They tend to be quite thoughtful. Yeah.
Thank you. Any questions from the audience? I think we just have time for one question.
The closest you can think of a really successful business model. It's not the only example. The one that I think works is Gilead example, where they take, there's a drug that for hepatitis C, as a very specific example, costs $1,000 a pill in the US, but the same pill they're willing to sell in places like India for $1. Yeah. I think at some point it's not about money. Seriously, I know, I'm saying it commercially. There is a... Yeah, I think companies do a lot of things. You call charity is one way to say it, but also I'll call it goodwill. People want to be associated with things that are force for good. I mean, I work in a completely for-profit company, but if I have a choice to support a biotech company versus a tobacco company, I always say, I have kids, I say, I never feel that whatever we do, it matters, despite the failure rates. Yatin and I were just talking earlier, our work, what we do, affects people's life every day. So I think for companies like Gilead, there were multifactorial points which don't have to be discussed here, but companies like India, unfortunately, sometimes they don't respect patent. So it is a way to mitigate all the business problems, create a value for the local country, and still have a goodwill, right? They don't give a patent to one company, they will give to 4 or 5 different companies. So it's a forced patent, but you are in return providing in a legitimate way the drug to the developing countries. Because frankly, you can't charge anyway the $1,000 pill model in India or Africa. Same thing Gilead did with HIV drugs. And that model seemed to be working for them, and there are other big pharma companies that have followed it. It's not purely for charity, but it's a way to avoid I call it distraction, because they generate their revenue in the US and developed markets, and they are still providing the service to the developing world. Nobody in their right mind wants to go to sleep thinking that people in India are dying because we charge $1,000 pill. And even if they would not want to care about it, countries like India provide the local company to violate the patents. There are multiple factors, but that's an example where things seem to work.
Yeah, well, I think your idea, that suggestion is pretty good, because if the data is collected and maintained properly. Because when I started my company, Medicinal, about 15 years back, we worked with some large groups in India, because India has a very large medical, chains, Apollo type of this thing. So in eye, for example, Narayana Netralaya, in diabetes, Dr. Mohan's, these are all like pretty spread out institutions with good data from a lot of longitudinal data from patients over 10 years, 15 years collected. But, I felt that, we should be able to monetize that to some extent, putting it all together into a database and trying to. But we found big challenges from the data of that time, because in the same institution, the data was not collected uniformly, the standards were not followed properly. So there are many challenges like that. But now, going forward, especially with something like Ayushman Bharat and things like that, if they plan and do it well, there's a very good chance that we will be able to get value from the insight. I don't know whether you'll be able to get discounted prices from drugs and all that, but there'll be definitely large AI companies and others like Google and all that who will be more than willing to come and take use of the data. Because even if 5 years back or 10 years back, Google was already there trying to work with many eye companies, many cancer companies trying to get data. So patient data will be very good. And then don't forget, it's not just patient data, human data. India is also very good. Many regions has got very good flora and fauna. Like the place I am from, Kerala. Kerala is one of the most biodiverse places in the world. So I convinced the Kerala government to start the Kerala Genome Data Center. So it took 3 years to get them to really get it approved and all that. Finally, the Kerala Genome Data Center is live, where we are collecting genomic data or generating genomic data of various plants, animals, livestock, fish, and all that, and storing it out in a government-owned database, and with a good AI, NVIDIA infrastructure also along with it. So we're trying to encourage startups to work on it. We're encouraging international companies to come and use the data, and we've not figured out a business model yet, but I'm hoping a business model will come out.
Yeah, thank you. Now, as we have heard the advances and new discoveries, as well as the challenges that exist in the process, I think prevention is the best way to maintain ourselves healthy. And at any of the panelists, at what stage of the treatment do we incorporate lifestyle interventions to help improve the quality of life of these patients?
So we do a lot of clinical work, so I'll try and answer the question here. Really challenging from a validation and proof perspective. And I'll use some specific examples. Specifically in oncology, there's a class of tumors called hypoxic tumors, which are suffering from lack of oxygen. And so depending on the metabolic pathways, there are specific therapies might work and specific nutritional regimens that might be helpful. Well, in oncology, most patients are outpatient. They don't stay in a hospital, so you can't quite monitor the nutrition habits and what they're feeding themselves. So anytime we try to run and consider a study in this where there's a specific drug that you are prescribing alongside a specific nutritional element, generally gets rejected not solely by the FDA, but even prior to that by the IRBs, who'll say it's rather impractical because you can't be quite certain what nutritional habits the patients will actually adhere to and comply with back in their homes. Certain other areas it might be easier. such as diabetes and weight loss and so on, because the tolerance may be a little better. But in oncology, most groups that eventually are going to validate the process don't find it feasible because patients are going to die.
Yeah. Thank you. I thank all the panelists for their diverse perspectives and contributing to this rich discussion. Thank you for your time.